Hi learners, I'm Dr. Gurunathan from Christu Jayanti College, and today we're going to discuss about a very important pathogen called Pneumocystis and its disease called as pneumocystosis. We're going to discuss about morphology and atypical fungal nature of this organism. To begin with, we are going to have an idea about what pneumocystosis is all about. It is also referred as pneumocystis pneumonia, commonly given as PCP. Sometimes it is also referred as interstitial plasma cell pneumoniae, and this is because there is intense plasma cell infiltration happening when this disease is present in human beings or other organisms. And this has been identified as an opportunistic pulmonary disease affecting humans and other broad range mammalian hosts, which causing atypical pneumonia. And the main region in the lungs that are affected is alveoli. And despite initially believed to be caused by a single protozoan species, based on some morphological features and drug sensitivity, now it has been recognized as a fungal disease based on nucleic acid and biochemical profile. The pathogen comprises a group of highly diversified species with an apparently strict host species specificity. The species that is known to infect humans is Pneumocystis girovesi. Formerly, this was called as Pneumocystis carinii. And the picture here is actually displaying you infection of lungs by Pneumocystis carinii. You can see that normal lungs are well, whereas when there is infection by this pathogen, right, you have a lot of disturbance in the lungs due to the alveoli getting affected. When you're speaking about few other important aspects related to this disease, it was first recognized in the mid 20th century as an important human disease, particularly as epidemics of interstitial pneumonia in premature infants and malnourished children in Europe. During 1960s and 70s, PCP was reported with increasing frequency in immunocompromised patients, particularly cancer patients, due because it was the time period where anti-inflammatory regimens were initially being developed, as well as intensive chemotherapy therapeutic aspects were introduced to handle cancer patients treatment. And only in early 1980s it became prominent that when a healthy young man was identified with pneumocystosis, they started understanding this disease to be a very important one. It is more common in immunosuppressed individuals, for example, HIV infected patients, cancer patients, and such immunosuppressed individuals, or patients suffering from immunosuppressed conditions. Whereas if it is an immunocompetent individual, it is usually asymptomatic or presents a mild respiratory infection kind of condition. And the incidence of PCP has been growing in patients even without HIV infection. And because of that, there has been a lot of special attraction to understand this disease. There has been multiple outbreaks, particularly among transplant patients, especially renal transplant patients. And colonization of the causative organism for this disease, pneumocystis, is also being identified in patients with a variety of pulmonary conditions, and it may be contributing to more rapid declines in pulmonary function. The most common symptoms when this disease is affecting humans are having a non-productive cough. The patients suffer from a low-grade fever. They also have dyspnea and chest tightness, and they are commonly having night sweats. Whereas in non-HIV patients or patients who have immunosuppressed conditions, the most common risk factors for this infection are suffering from asthma, suffering from cystic fibrosis, suffering from systemic lupus erythematosus, pregnancy conditions, rheumatoid arthritis, and ulcerative colitis. Along with all these things, people who are having chronic obstructive pulmonary disorder, that is COPD, they also tend to acquire pneumocystosis infections very easily. And when you speak about the causative agent, it is a unicellular atypical fungi which is classified based on their phylogenetic information into the Ascomycetes group. And you can see that they have a lot of common characteristics with fungi, but still it is considered to be atypical fungi because it has substantial differences from the existing fungi we know. The knowledge of the atypical nature of pneumocystosis is evolving gradually with more advances in the pneumocystis research currently happening. A major atypical feature of this organism is it has a pleomorphic shape and it has a fragile cell wall, which is in contrast to the presence of a rigid cell wall in typical fungi. Not only that, this is the only fungi which lacks chitin in its cell wall, whereas all the other fungi that we know till date have chitin or related glycoproteins, mannins, glucans, chitins, and chitosans in their cell wall, whereas pneumocystis doesn't has any of these materials. Interestingly, they have also identified that enzymes which are required for the biosynthesis and degradation of beta-1,3 glucan and beta-1,6 glucan are present in this organism, whereas the enzymes that are required for the biosynthesis as well as degradation of alpha glucan molecules is not present in this fungi. This is again a very interesting identification factor of this organism. And the third atypical feature of this organism is their inability to propagate the organism in vitro. Even though we have fungal culture media and other culture systems available for cultivating a regular fungal member, it's not so easy to cultivate pneumocystis in a laboratory setup. And whole genome analysis provides some possible insights into this in the current scenario. And compared to those of other fungi, the genome that is present in Pneumocystis is highly compact, and that makes the organism to be highly dependent on the host to complement these small, say, genome that is present there. As the genome is smaller, it would have definitely lost a lot of genes, and to compensate that, right, okay, it goes on with these activities. And another important feature that they have identified with pneumocystis is it is also co-evolving with its host species. And because of that, there is exclusively infecting ability only on one particular host species and not having the ability to infect many different hosts. So there's again a very interesting atypical nature of this organism that makes this organism an interesting member to study. When you speak about the morphological aspects of this pathogen, they are existing in three distinct morphological forms. One is called as the trophozoites, the other one is pre-cystic form, and the last one is cystic form. In trophozoites, they are generally irregularly shaped, they are thin-walled, which is usually 1 to 5 micrometer in size, and they reproduce by binary fission. The picture that is shown there is actually a schematic representation of the trophozoites that are present there. You can see that on the surface of the organism, you have tubular expansions, which helps the organism to actually attach with intestinal cells when they are infecting humans and other organisms. When you speak about pre-cyst, this is considered to be an intermediate stage in sexual phase of reproduction, which is leading to the cyst formation of the organism, and usually it has 5 to 8 micrometer size. And in cystic phase, you can see that the organism is spherical in nature. It is thick-walled and it is usually having 8 micrometer size, and it generally harbors up to 8 intracystic bodies. And the cyst collapses to release trophozoites, and these trophozoites can initiate another cycle of multiplication either in the same host or in a different host. Collapsed cysts observed as irregular concentric bodies, and the picture that is shown here is a typical schematic representation of this organism. With this, we would conclude our session today. We'll meet in another interesting topic later on. Take care and thank you for your patient listening.